Hello, and thank you for joining us. I'm Gwen Paler, Senior Developmental Editor with Current Protocols at John Wiley & Sons, and I'm delighted to introduce today's webinar titled Measuring Toxicity Biomarkers in Mouse Models and Other Small Volume Samples. This webinar is being co-sponsored by Current Protocols and EMD Millipore. EMD Millipore is the life science division of Merck KGaA of Germany and delivers products and services for life science research in academic and pharmaceutical environments. They offer the largest portfolio of biomarker assays, including ELISAs, radioimmunoassays, and multiplex assays that enable researchers to understand complex biological systems and identify new diagnostic and therapeutic targets. EMD Millipore is a preferred distributor of the entire Luminex portfolio of multiplexing instruments, and their multiplex panels are manufactured to industry gold standards, giving users the same accuracy and precision in every lot, backed by a highly knowledgeable technical support scientist. Current Protocols has been in continuous publication for over 27 years and is the largest collection of peer-reviewed, authoritative, and regularly updated step-by-step research techniques and procedures available for life scientists worldwide. With 17 titles and over 16,000 protocols, Current Protocols is part of Wiley Publishers. During today's program, we encourage you to submit your questions throughout the event by clicking on the Ask a Question box at the bottom of your screen. Your question will not be seen by any of the other attendees, so please don't be shy about asking them. The webinar will be recorded and available for viewing within the next few days. We will send you an email with details on how to access the recorded webinar, along with a PDF of the slides and a customizable certificate of attendance. In addition, today's viewers will be able to download a complimentary copy of a white paper from EMD Millipore on kidney toxicity biomarker analysis. I'll give you details on how to obtain this white paper at the end of the webinar. But right now, it's my pleasure to introduce today's speakers. Dr. Mitch McGill is a postdoctoral fellow in the laboratory of Hartmut Jaschke at the University of Kansas Medical Center. He has worked in the field of biomarker research for the last 5 years and is building a reputation as a leader in the development of novel serum biomarkers of liver injury. He has authored several book chapters and more than 30 research articles, including papers on liver injury biomarkers. Dr. Venkata Sabbisetti is an instructor in medicine in the renal division of Brigham and Women's Hospital at Harvard Medical School. Dr. Sabbisetti has a broad background in kidney pathophysiology and biomarker discovery and analysis. He has over 10 years of experience in clinical translational science and over 6 years of experience in biomarker studies. He is a member of the Chronic Kidney Disease Biomarkers Consortium and has published numerous peer-reviewed publications and book chapters on biomarkers in kidney diseases. So let's get started with a very warm welcome to you, Dr. McGill.
Thank you, Gwen, for that introduction. So today, I'd actually like to start this presentation. I'll be talking a little bit about laboratory methods. I'd actually like to start this presentation, though, by sharing my own experiences using biomarkers in research, because it will help me to illustrate a few points that I think really need to be highlighted in this area. So my focus of research is liver injury, and in particular, drug-induced liver injury, or DILI. This is, of course, a major problem at every step along the way of the new drug pipeline, from initial experimental studies through clinical trials, and even after a new drug has hit the market. And if it's encountered at any one of these stages, the new drug will likely be killed right then. In fact, until the introduction of new biomarker-based scores to identify the potential for hepatotoxicity, drug-induced liver injury was the most common cause of post-marketing drug withdrawal. And it is still, according to my understanding, the most common cause for drug failure during pre-marketing trials. Now, there are actually two types of DILI. There's idiosyncratic DILI, which is unpredictable, it's not dose-dependent. It can occur even after many months of asymptomatic use of the causative agent. And then there's intrinsic DILI, which is the more traditional dose-dependent toxicity that we usually think of. And this is much more predictable. Unfortunately, idiosyncratic toxicity is very difficult to study because it's so unpredictable. However, intrinsic toxicity is quite convenient and easy to investigate, which is great because the most common cause of drug-induced liver injury in this country is actually an intrinsic hepatotoxicant. and that drug is acetaminophen. So acetaminophen, of course, is a very popular analgesic. Actually, it's the most used drug in the United States. But overdose can cause severe liver damage. In fact, it is the greatest single cause of acute liver failure and ALF-related deaths in this country. So the mechanisms of acetaminophen hepatotoxicity begin with conversion of the drug to a reactive metabolite that binds to proteins. And binding to mitochondrial proteins in particular causes mitochondrial dysfunction and oxidative stress. And the oxidative stress then activates multiple kinase signaling pathways that converge on the C-Jun N-terminal kinase, or JUNK. And JUNK can then translocate into mitochondria where it enhances the mitochondrial oxidative stress. And eventually, you get the occurrence of the mitochondrial membrane permeability transition with ATP depletion, and swelling and rupture of the mitochondrial membranes results in release of mitochondrial content, including some endonucleases, into the cytosol. Those endonucleases can then translocate into the nucleus and fragment nuclear DNA. And the result of this sequence of toxic events is oncotic necrosis of hepatocytes in the liver. Now, this is what we've known in mice for many years. However, very little has been done until recently to translate these mechanisms to humans. So we decided to fix this in my lab, starting with the role of mitochondria in humans. Unfortunately, it's very difficult to obtain tissue from acetaminophen overdose patients. So a major question that we had to ask ourselves was, how do we study detailed mechanistic events occurring in tissue using only blood? And this is what we came up with. So we reasoned that if mitochondrial damage and rupture occur in humans as they do in mice, then the mitochondrial content will spill into the cytosol and should become detectable in plasma when cell death occurs and plasma membrane integrity is lost. And consistent with that idea, we were able to show that several mitochondrial markers were dramatically increased in the circulation of patients with acetaminophen-induced liver injury. For example, the mitochondrial matrix enzyme glutamate dehydrogenase, or GDH, and mitochondrial DNA were both increased in plasma from acetaminophen overdose patients with liver injury, referred to here as the abnormal liver test results group, as compared to overdose patients with normal liver test results and to healthy volunteers. We also observed an increase in nuclear DNA fragments, which requires mitochondrial damage in mice, so we consider it a sort of indirect biomarker for mitochondrial damage in the serum. And just as a side note, measurement of these nuclear DNA fragments is based on capturing histones in the DNA, so it's not actually a measurement of the DNA itself. Now, interestingly, for the kidney people in the audience, Rick Schnellman's groups from South Carolina presented very similar data at the Society of Toxicology meeting earlier this year, showing that urinary mitochondrial protein and DNA levels are elevated after acute kidney injury. But a key point here is that most of the popular and emerging biomarkers right now are macromolecules. So we're looking a lot at proteins and nucleic acids. So a good biomarker platform will be able to detect these types of analytes. Presently, there hasn't been much use of metabolomics approaches in the field. So although you'll see that on some of my later slides, I'm not really going to discuss it in any detail. And this is likely because of the cost and limited availability of the necessary equipment. So most of the popular biomarkers right now are proteins and nucleic acids. Now, getting back to the data, of course, it's easy to imagine that if you have massive cell necrosis, you will likely get some release of mitochondria or mitochondrial markers into the circulation, simply as a secondary effect. So it was important for us to ask, are these biomarkers really specific for mitochondrial damage, or are they just coming out as a secondary effect of the necrosis? And to answer that, we treated mice with either acetaminophen or with a single large dose of the diuretic furosemide, abbreviated FS here. Fortunately for us, a group in Canada had previously shown that furosemide actually causes a very similar pattern of centrilobular liver injury, but it does so without affecting mitochondria. So we decided to use furosemide as a sort of negative control for mitochondrial damage. And when we did this, we found that GDH and mtDNA were only elevated in serum from the acetaminophen-treated mice. So we think that this is at least some evidence that these biomarkers are, in fact, specific for mitochondrial damage. Now, the furosemide model worked brilliantly for our purposes. However, one problem that we encountered in these experiments was that the furosemide, of course, as I mentioned, is a diuretic. That's actually its clinical application. And giving a large dose of a diuretic means that these animals urinate quite a lot and become dehydrated, so the plasma sample volumes that we were able to get were quite low. And small sample volumes is a challenge that we have occasionally faced in the clinic as well with patients from whom it's difficult to obtain blood due to small veins or rolling veins or other common problems. So another key point is that a good biomarker assay needs to work even with small sample volumes. Now, at this point, we knew that mitochondrial damage occurs in acetaminophen overdose patients from these measurements, but that doesn't mean that it has a critical role in the pathogenesis. Of course, we can't give these patients whatever intervention we like to prevent the mitochondrial damage, so in order to test our hypothesis that mitochondrial damage is actually important, we decided to compare the levels of these mitochondrial damage biomarkers in the serum of non-survivors designated NS versus survivors designated S of acetaminophen-induced acute liver failure, figuring that the mitochondrial damage biomarkers would be higher in the serum of the non-survivors if it really is critical. And sure enough, that's exactly what we saw. While ALT, alanine aminotransferase, which is the standard serum marker of liver injury that's used clinically, was not elevated in the non-survivor group, all of our mitochondrial damage biomarkers were. So this actually led us to wonder if these biomarkers might be clinically useful for the prediction of patient outcome. And to test that possibility, we performed a receiver operating characteristic curve analysis. And interestingly, all of our mitochondrial damage biomarkers could predict patient death in this cohort, while ALT could not. So all of them had AUC values that were higher than 0.5, were close to 1, and p-values that were less than 0.05. However, the sensitivity of these biomarkers was pretty poor, and the predictive power was not as great as we would have liked to see. The AUC values were always between 0.7 and 0.8, and we'd like to at least crack that 0.8 line. So we decided to try combining all of these biomarkers into a single score, which we call the Mitochondrial Damage Biomarker Index, or MDBI, on the right side here. And as we hoped, we found that combining the markers at least modestly increased the AUC. And surprisingly, it actually performed somewhat better than the Model for End-Stage Liver Disease, or MELD score, which is another score that is already used clinically to predict the need for a liver transplant. And perhaps more striking, the odds ratio for the MDBI was actually much higher than that for MELD. So another key point here is that multiple biomarkers are better than one. So a convenient platform for biomarker monitoring would allow the measurement of multiple markers at the same time. So you really want the assay to be as high throughput as possible. This is just a summary of those key points. I think, in my opinion, a good biomarker platform or assay will be used to detect macromolecules. It'll work with small sample volumes, and you'll be able to measure multiple analytes in multiple samples in a single run. So these are just the probably the most common techniques used to measure biomarkers in body tissue and fluids, both clinically as well as in the research lab. And I'll just run through this, briefly run through each one. So one approach that I personally use quite a bit, and it's used frequently to measure liver injury biomarkers like ALT and AST in the clinic, is enzyme kinetics. So many of the standard clinical markers of liver injury, as well as many emerging biomarkers such as argininosuccinate synthetase and carbamyl phosphate synthetase, are enzymes. So they can be measured by quantifying their activity in serum using a kinetic assay. And this is generally done by mixing the sample in a buffer containing substrates and co-substrates that are specific for the reaction catalyzed by the enzyme of interest. and then tracking the disappearance of one of the substrates by monitoring loss of absorbance at the substrate's absorbance maximum. That's a great approach because it's relatively cheap and it's very accurate when it's done correctly. However, unless you have access to a plate reader, this can be a time-consuming and laborious process. And even with a plate reader, you can generally only measure one analyte at a time, and often even only one sample at a time with a standard spectrophotometer. Often these enzymes actually don't have very high intrinsic activity, and they may be difficult to detect without extremely long reaction times. So the sensitivity is a little bit low in some cases. So perhaps a better way to measure proteins, including enzymes, we actually recently used this approach to study arginine succinate synthetase as a biomarker of liver injury, is to do an ELISA. So this is an enzyme-linked immunosorbent assay. For the uninitiated, there are a number of variations on this, but the basic idea is that you bind the antigen of interest to an antibody in such a way that both are immobilized on a solid surface, usually in a 96-well plate, and then detection reagents are added, and in the case of a sandwich ELISA that's illustrated here at the bottom right, a capture antibody against your antigen of interest is immobilized on the bottom of the plate. Your sample is added, the antigen binds to the antibody, and you can then add a secondary antibody, which can have its own reporter of some kind, usually fluorescent label, or which can be targeted by another antibody with an enzymatic reporter. And then you can measure it either as a kinetic assay, in the case of enzyme-linked immunosorbent assay, or if your reporter is just a fluorescent reporter, then you can measure it as an endpoint assay. So the advantages of this approach include much greater sensitivity and the ability to run multiple samples in a single run. However, most ELISAs are designed to only measure a single parameter or analyte at one time. Of course, another option is mass spectrometry. Obviously, this is a very robust approach. The number of analytes and samples in a single run is almost unlimited. However, the disadvantages are not trivial, to say the least. It requires very expensive equipment and a considerable amount of expertise to run that equipment. And if you're using serum, you'll likely also find that you need to deplete your samples of albumin, immunoglobulins, and possibly a few other proteins that together account for more than 50% of serum protein. So a problem that we regularly see is that the overwhelming presence of these few proteins can interfere with detection of the proteins in which we are interested. And these depletion protocols typically involve the use of 10 or more antibodies. So this can really drive the cost of each sample into the hundreds of dollars, and it's really very time-consuming. Now, for nucleic acids like mitochondrial DNA, you can use real-time PCR. Here again, you can measure multiple analytes and run multiple samples in a single run. However, isolating and concentrating the DNA can be fairly laborious, and in my experience, it can actually require relatively large volumes of urine or serum just to obtain a sufficient amount of material. So we're talking about anywhere from a couple hundred microliters to as much as 1 milliliter of sample. Of course, other options for nucleic acids include microarrays. In general, these approaches have the same pros and cons of RT-PCR, but with the added disadvantages of greater cost and the need, again, for expert assistance. And the same is generally true of sequencing. You can run a lot of samples. You can measure pretty much all of the nucleic acids in the sample. However, again, it's very expensive and requires a lot of expertise. Now, another approach that does not suffer from many of the disadvantages of these other methods, and which I think is really going to be the focus of Dr. Sabbisetti's talk, is bead-based multiplexing. So in these assays, you use different unique sets of beads. Each set has its own spectral or size properties that can be used to distinguish one from the other. And each set is conjugated to a different primary or capture antibody. And in this way, you can probe for multiple analytes in a single aliquot of a sample and tell those analytes apart by the unique properties of the bead to which they're bound. So the basic procedure is illustrated here on the right. You mix your sample in with these beads. The capture antibodies on the different beads bind to their respective antigen and the sample, if it is present. You then wash away the unbound antigen and then add a secondary antibody with some kind of reporter system, and then run the samples through, in some cases, a flow cytometer or often another similar proprietary system. And basically, you simultaneously measure the signal from both the bead and the reporter as it flows through. And in that way, the instrument can tell which primary antibody resulted in each positive reporter signal. So this method really has all of the things that I would look for in an ideal assay. Allows you to measure multiple analytes and multiple samples in a single run. Fairly small sample volumes can be used in general, and it really doesn't require much special expertise or training. The basic procedure is very similar to an ELISA, just with a little different detection method. And finally, I believe that both proteins and nucleic acids can be measured, although not necessarily on the same plate. So these are some examples of potential analytes. So for example, you could potentially set up an assay in which you measure argininosuccinate synthetase, a very sensitive and early serum biomarker of hepatic damage, with the caspase cleaved form of keratin 18. And that way you could get an idea of when and how much cell death is occurring and whether or not apoptosis may be involved. And I think that this technology actually really lends itself to cytokine analysis, because you typically want to measure multiple cytokines at once to get a clearer picture of what's happening. And in fact, our lab has actually done this in serum from acetaminophen overdose patients, and I'll just briefly share those results with you. Surprisingly, we actually found that most of the cytokines that we measured do not change during or after acetaminophen-induced liver injury. And this is interesting to us because there's a popular line of thought that says that innate immunity, sterile inflammation with damage-associated molecular patterns, is actually a critical part of acetaminophen toxicity, and the IL-1 beta in particular is important in this. But as you can see, IL-1 beta, like the other cytokines, really did not increase at all. So this technique has allowed us to use biomarkers to really better understand acetaminophen-induced liver injury in humans and to really test a major hypothesis that has come from rodent data. So I'll just quickly acknowledge some of the people who have helped me out with this study, particularly my mentor, Dr. Yeshke. I also, again, like to thank Gwen for the opportunity to share this with you. And I'll go ahead and turn it over to Dr. Sabbisetti.
That's a wonderful presentation by Dr. McGill. As he pointed out that liver is one of the primary organs of toxicity. And in the similar fashion, kidney is one of the primary organ of drug toxicity. And actual renal injury is the second leading cause of drug attrition after liver toxicity. However, there is only limited ability to monitor kidney damage associated with the drugs that have been in use. So today in my presentation, I have been focusing on several novel biomarkers and the novel methodologies that have been developed recently which enables measuring multiple biomarkers in small quantities of urine. So even though kidney is a widely assessed organ during preclinical safety testing, owing to its role in the drug excretion and detoxification process, nephrotoxicity accounts for failure of only 7% of new drug entities. On the contrary, drug-induced acute kidney injury in the intensive care setting accounts for approximately 30 to 40% of cases. This lack of correlation indicates the underestimation of nephrotoxicity assessed by routinely used biomarkers during preclinical studies. Though histopathology still remains to be the gold standard to identify nephrotoxicity in animal models, this approach will only facilitate the assessment of kidney injury post-mortem. Unfortunately, the routinely used gold standard biomarkers like serum creatinine to assess renal toxicity is very problematic. The reason why I mention this is problematic because the serum creatinine levels varies widely with age, gender, diet, muscle mass, and several non-renal parameters. Further, in order to see a significant change in serum creatinine, one has to lose 50% of kidney function because of the presence of renal reserve. So serum creatinine can take several days to reach a new steady state. So what role does biomarkers play in kidney research? Firstly, biomarkers can enable early diagnosis at clinically manageable stage. And kidney is a heterogeneous organ. So a biomarker which is specific for one cell type or which reflects injury to one cell type may not enable or may not reflect the kidney injury accurately. Thus, employing multiple biomarkers which reflect multiple sites of injury is crucial in assessing the kidney injury. Further, biomarkers enable to differentiate diseases of various etiologies and can predict outcome. And we recently demonstrated that blood KIM1 levels in diabetic patients at baseline predicts progression of diabetic nephropathy to end-stage renal disease. And also, these biomarkers ultimately expedite the drug development process. Over the past decade, novel renal injury biomarkers that are more specific and sensitive than serum creatinine were discovered and validated in rodent and human studies. Recently, FDA and European Medicines Agency have qualified some of these biomarkers, namely KIM1, albumin, total protein, beta-2 microglobulin, cystatin C, cholesterol, and trefoil factor 3 to monitor kidney toxicity in preclinical studies. Because of the ability to genetically modify the mouse has become very important animal in understanding the injury and repair mechanisms of the kidney and characterizing nephrotoxicants. However, the use of these novel biomarkers in drug development has been hampered by the lack of sensitive, specific, and robust assays that enable measurement of these markers in small quantity of biological specimen in mouse models of renal injury. As Dr. McGill pointed out in his presentation, that using conventional assays, one can only measure one biomarker at a particular time in a single well. So this requires larger volume of biological specimen. Thus, assays that enable measurement of multiple biomarkers simultaneously in a single well is crucial when dealing with small quantities of biological specimen, especially in preclinical setting. And microbead-based assays, which are measured on Luminix platform, enables measurement of multiple biomarkers at a single time using small quantities of biological specimen. So recently, Milliplex has developed several kidney toxicity biomarker panels to measure multiple biomarkers in urine and serum specimen. And as you see in the slide, there are 4 panels, and the panels which were highlighted in blue are intended to measure multiple biomarkers in urine, whereas panels 3 and 4 are intended to measure biomarkers in serum. And these biomarkers are segregated based upon their dynamic range in this specimen. And in panel 1, there are 6 biomarkers, namely KIM1, TIMP1, IP10, VEGF, renin, and beta-2 microglobulin. And in panel 2 contains NGAL, cystatin C, cholesterol, EGF, and osteopontin. Panel 3 contains renin, KIM1, TIM1, IP10, VEGF, and panel 4 contains beta-2 microglobulin, cystatin C, NGAL, cholesterol, and osteopontin. So as a first step, using these panels, we generated standard curves to understand their dynamic range. So we diluted the standard to several dilutions, and we generated a standard curve, and we performed a logistic regression model to fit these curves. As you see, all these standards, all these biomarkers have generated a nice standard curves, and the upper and lower limit is according to Milliporkit's recommendations. We evaluated intra-assay variability of these biomarkers using these panels by measuring 20 samples of urine collected from mice that are exposed to kidney injury, and each marker is measured in quadruplicate. And as you see in here, the coefficient of variation for biomarkers in the panel 1 is below 10%, and in panel 2, the coefficient of variation is below 13%. Using these assays, we sought to explore the time course of changes in serum and urine levels of these biomarker panels, along with serum creatinine in 2 mouse models of renal injury. In the first model is ischemia reperfusion injury, in which the mice were subjected to 30 minutes of bilateral renal ischemia, followed by 6 to 120 hours of reperfusion. In the second model, mice were subjected to 1 intraperitoneal injection of cisplatin at 20 milligrams per kg dose, and followed at several time periods. As I pointed out here, we used biopsy male mice, and we performed renal ischemic perfusion injury, where we clamped both renal pedicles for 30 minutes, and the mice were maintained at 37 degrees Celsius. And after 30 minutes, we unclamped the renal pedicles and we allowed the reperfusion, and we followed the mice for several time periods, and we collected the serum, urine, and tissues and kidneys at several time periods. As you see here, we measured serum creatinine, and following renal ischemic perfusion, it took 12 hours to see a significant change in serum creatinine as compared to sham-operated mouse, where the kidneys were exposed but not clamped to serve as controls. And serum creatinine levels were significantly elevated only 12 hours after severe kidney injury, and the levels were remain elevated till 72 hours. Whereas the novel biomarkers, including beta-2 microglobulin, renin, and other markers, behaved differently as compared to serum creatinine. Beta-2 microglobulin took 24 hours to see significant elevation as compared to sham, while renin, KIM1, TIM1, and IP10 were elevated much more early than serum creatinine. Especially KIM1, IP10, and TIM1 were elevated within 6 hours after the bilateral ischemia. The levels of KIM1 were sustainably elevated till 120 hours, as demonstrated in rat models of kidney injury, and TIM1 levels came to almost normal by 72 hours, and IP10 levels came back to normal range by 120 hours after reperfusion. So all of these biomarkers have different kinetics. And we also measured panel 2 biomarkers, which contain cystatin C, EGF, NGAL, cholesterol, and osteopontin. And cystatin C was elevated within 6 hours after bilateral ischemia, whereas EGF levels were significantly downregulated or decreased within 6 hours after ischemia reperfusion injury, and the levels were consistently down even after 120 hours after ischemic reperfusion. Again, NGAL clustering have outperformed serum creatinine, and they both are elevated within 6 hours after the ischemic reperfusion injury, whereas osteopontin was elevated by 12 hours. And one important aspect here is that these biomarkers have huge dynamic range, and the levels are very high in the serum. So to fit these biomarkers in the dynamic range of the assays, it requires 1 in 400 sample dilution. So you require only very little sample specimen to measure these biomarkers. So 1 microliter of sample can be diluted to 400 microliter, and you require only 25 microliter to measure in single case for all these biomarkers. As a second model of renal injury, we performed drug-induced kidney injury, and cisplatin is a very well-known nephrotoxicant. To induce kidney injury, we gave 1 intraperitoneal dose of cisplatin at 20 milligrams per kg dose in biopsy male mouse, and serum and urine and tissues were collected at 24 hours, 48 hours, and 72 hours to measure biomarkers. As shown here, there was no change in serum creatinine at 24 hours following 1 dose of cisplatin. However, there was significant elevation of serum creatinine within 48 and 72 hours as compared to mice that were treated with vehicle alone. And we measured novel biomarkers using the 4 panels developed by the Milliplex, and there was no significant change in beta-2 microglobulin following cisplatin injury at any given time period, and renin was significantly elevated by 72 hours, and similarly TIMP1 is also elevated by 72 hours, whereas KIM1 and IP10, there was a huge increase within 24 hours following cisplatin mediated kidney injury. Similarly, VEGF also took 72 hours to see a significant elevation in the levels. So from this slide, it can be clearly interpreted that KIM1 and IP10 are much more sensitive in detecting cisplatin-induced kidney injury as compared to beta-2 microglobulin, renin, TIM1, and serum creatinine. In panel 2, again, cystatin C and EGF outperformed serum creatinine. Cystatin C is increased within 24 hours following the injury, and EGF was decreased significantly within 24 hours following the injury. Whereas NGAL was, it took 48 hours to see a significant rise in NGAL levels as compared to vehicle treated mice, and in clustering, it took 72 hours to see an increase in the biomarker levels. So both cystatin C, EGF in panel 2 and KIM1 and IP10 in panel 1 outperform serum creatinine and other biomarkers. So then we moved on to panel 3, which is mainly intended to measure biomarkers in serum. Again, we performed standard curve experiments to see the dynamic range and to assess upper limit of quantification and lower limit of quantification of these biomarkers, and they're all performed as recommended in Milliplex or Milliplex kit directions. And we measured intra-assay variability of these panels, and as you see here, panel 3 contains renin, KIM1, TIMP1, IP10, and VEGF, and intra-assay variability is less than 10%. And in panel 4 also, which contains beta-2 microglobulin, cystatin C, NGAL, cholesterol, and osteopontin, the intra-assay variability is again less than 10%. So they do have very tight intra-assay variability. And again, the intra-assay variability was assessed by measuring 20 serum specimens, and they measured in quadruplicate for all these biomarkers. And these are the standard curves for panel 4, and they show an excellent dynamic range with very nice upper limit of quantification and lower limit of quantification for each biomarker. So we utilized the specimens which were collected from the Ishmael perfusion experiment to measure these biomarkers. And as shown here, we measured, we diluted 1 in 5 for panel 3, and we measured renin, TIMP1, IP10, VEGF, and KIM1 in the serum specimen. And as compared to the serum which was collected before the injury, the levels of TIMP1, IP10, and KIM1 were significantly elevated within a day after the perfusion injury, whereas there was no significant change in the serum levels of VEGF and renin were observed following Ishmael perfusion injury. In panel 4, there was no change in the levels of beta-2 microglobulin, cystatin C, or cholesterol following Ishmael perfusion injury, whereas there was significant elevation of NGAL and osteopontin within 1 day after the Ishmael perfusion injury. These indicate that all these biomarkers have different kinetics, and they reflect different parts of kidney injury. So to conclude my seminar, novel biomarkers are sensitive and early as compared to serum creatinine, and multiplex panels enable to measure multiple biomarkers in small quantities of sample specimen, which is crucial in preclinical setting when dealing with small quantities of biological specimen. Thank you very much. I'm happy to answer all your questions.
All right. Thank you, Dr. Sabbisetti. Now, please welcome Dr. Steve Suchta from EMD Millipore. He has some information on how you can learn more about the array of toxicity biomarker panels that are available for bead-based multiplexed assays. Steve?
And I'd like to thank the speakers for both of their great webinars as well. I'd like to just briefly talk to you about some of the Milliplex MAP kidney toxicity panels. Dr. Sabbisetti actually briefly mentioned these as well. We've taken great care into building our toxicity panels. One thing I wanted to mention is that the way we kind of build up the biomarkers that go into our panels is through using the guidance of the Predictive Safety Testing Consortium, the PSTC. We kind of use that as our guidance for building the panels, and then kind of using the advantage of multiplexing, putting in different exploratory biomarkers in those same panels. But we always go back to those tested biomarkers that have been approved by the PSTC. So briefly, I just wanted to mention some of there are 7 PSTC qualified urinary kidney injury biomarkers. One of them is urinary total protein, which isn't included obviously as part of our panels, but there are the 6 additional qualified level of biomarkers, and qualified is essentially the highest level of investigation that the PSTC has done. And as you can see from this table, for all of our panels, we essentially have all 6 of the qualified urinary kidney injury biomarkers, except for TIF3 is missing from the rat panel. The other level of urinary kidney injury biomarkers is research level, and that's kind of the second level down from the third level of investigation from the PSTC. And again, we use these as a way to kind of build our panels based on these and then build in additional biomarkers. And you can see here that, again, we have, but there are the 5 research kidney injury biomarkers, and you can see here from the rat, canine, and human that we have several, most of these biomarkers are contained in the panels. And then finally, I'd just like to mention that all this information about all of our biomarkers, all of our analytes can be found in our Analyte Quarterly. And it's available online at the emdmillipore.com/Milliplex. And actually, we have things like cytokines that were mentioned, cancer biomarkers, as well as the wide array of toxicity biomarkers. And we also just recently launched a rat liver toxicity biomarker panel. And with that, I'd like to hand it back over to Gwen.
All right, thank you, Steve. So let's now proceed to the question and answer segment. So if you haven't yet submitted a question, now is the time to do so by clicking on the Ask a Question box at the bottom of your screen. Also, we have a special offer for attendees at the end of this webinar, so stay with us to get the details for that. So let's see what questions have come in so far. This one looks like it could be for both of our primary presenters. What sources do you use to look for novel biomarkers? Mitch, you want to maybe take a start at that?
Sure. So there's kind of two approaches that we've taken. One is just really kind of hypothesis-driven research. Like, for example, with those mitochondrial biomarkers, we just thought about the mechanisms of acetaminophen toxicity that are known from rodent studies and how that develops, and just went from there and said, okay, what can we measure in the plasma that might be coming out of these mitochondria? So in that sense, it really wasn't like we had done any preliminary studies or anything. We just kind of went with what was already known, formed a hypothesis and went with that. In other cases, we use more sort of high-throughput approaches, where doing proteomics or metabolomics studies initially in rodents, and then going in and exploring, picking out individual biomarkers from those data that look promising, and then investigating those further. But I think a similar thing could be done, for example, with the multiplexing technique, where you can look at panels that might be available from the company or assemble your own panels, and then kind of pick out from there individual proteins or other biomarkers, potential biomarkers that change, and further investigate those.
Okay, Dr. Sabbisetti, what about you? What sources do you use to look for novel biomarkers?
I completely agree with Gwen. We look into tissues in the beginning, and then we do proteomic approaches, either in urine or plasma, and we cross-compare them with the tissue levels. Some of this might originate from the tissue, especially in kidney injury. Either the changes in the biomarkers could be coming from the changes in the immune system, or could be actually shedding from the kidney cells. So the plasma would enable us to measure not only the biomarkers which are actually coming directly from the kidney, but as a change in the whole body immune function reflecting kidney injury. So there, in those cases, plasma would really serve as identifying novel biomarkers. For instance, recently we have been looking at a complement factor, which came by looking in the plasma after a kidney injury. But we couldn't see that in the tissues, much of staining in the tissues. So I would go with both tissue and biological specimen, whether it's urine or plasma.
Okay, our next question also can be answered by both of you, I'm sure. The question is, what are the main issues when making the translation of results in animal models to human samples? So Dr. Sabbisetti, perhaps you first this time?
So the main issue we will see, for instance, I can highlight KIM1, which I work a lot. So if you look at KIM1, the levels change dramatically in animal models after ischemic repair fusion injury. So the fold change is almost 700 to 800 fold as compared to the animals with no injury or before the injury. So that fold is significant enough to predict that you might see a significant change in humans, and also whether the biomarker is well conserved, or is it, for instance, again, KIM1, you have KIM1, KIM3, and KIM4 in humans, whereas you miss some of the KIM components in mouse. So, but KIM1 is well preserved across species. So that is one. The second thing is the technology. Before it translatable, the technology must be very robust to measure the analytes. If you don't have an assay which is not robust, then it would be very hard to translate the studies into humans.
All right, over to Mitch. Question, same for you. What are the main issues when making the translation of results in animal models to human samples?
So I think, I guess I actually don't have much to add to Venkata's answer. I thought it was a pretty good response. So essentially, there's always this consideration of species differences. Almost no matter what you're measuring, you have different isoforms of proteins. Some proteins may not be expressed in rodents that are expressed in humans, or vice versa. And even if they are the same isoforms, they may not be recognized by, for example, if you're doing an ELISA, they may not be recognized by the same antibody. Another challenge or consideration, of course, that you kind of have to consider, I think, is maybe the two things. For example, if you're studying a disease, maybe the disease just doesn't occur the same way in the rodent model as it does in a human. So you may not see the same type of injury or the same mechanism of injury, which could result in differences in what biomarkers are released or detectable in either urine or plasma. So, for example, a lot of people use mice to study alcohol-induced liver injury in my field. However, mice really don't respond to alcohol in the way that humans do. They don't develop the same kind of fibrosis and cirrhosis. So certainly, in addition to the issues that Venkata mentioned, you may have to consider that some models, just like in all science, some models may not be all that good for the human situation.
Okay, this next question I think is for you, Dr. McGill. It's how do you differentiate between biomarkers coming from damaged and ruptured mitochondria and those from intact mitochondria that might be released into the serum?
Yeah, so I think that's an important consideration for anyone who's interested in these mitochondrial damage biomarkers. The way that we do this, and this has actually become the standard protocol for the Drug-Induced Liver Injury Network, which is a national network that's devoted to the study of drug-induced liver injury, is we actually spin down the plasma, almost like a differential centrifugation protocol. We get it fresh from the patients and then separate it to obtain the plasma or serum, and then we spin that down at very high forces to remove any intact mitochondria, just pellet those down, and then take the supernatant off.
Okay. I think this is for Dr. Sabbisetti. This question is, what about saliva as a source of biomarkers of kidney disease? Is anything known beyond urea and creatinine?
Oh, that's an excellent question. Actually, no, we actually never looked into saliva for kidney diseases. Maybe we may find definitely some markers, because I looked into saliva as a specimen when I was looking for biomarkers for endometriosis. This is one of the gynecological conditions in women. But in kidney injury, I haven't seen many publications actually focusing on saliva. Mostly, most of the research mainly focused on urine, because there's immediate product you can find filtering through kidney where you can find abundance of biomarkers. So actually, I haven't looked into the saliva part yet in kidney diseases.
Okay, this one I think is up for grabs. So whoever wants to take it first can. This one says, have any stability studies been performed on these biomarkers?
It's a good question. In my case, the biomarkers that I presented here, other than GDH, which had been studied actually decades ago as a separate marker of liver injury, but not specifically with a focus on mitochondria, none of them have really been tested thoroughly for that. It's preliminary, pretty preliminary in the early stages.
And Dr. Sabbisetti, what about the biomarkers that you have studied? Have any stability studies been performed on those?
Yeah, definitely. So most of these biomarkers actually approved by FDA and to use them in preclinical studies. So we have done extensive studies to understand the stability of these biomarkers, both in mouse and in human, because often in clinical studies, there is always a question like how long you can keep the sample specimen at room temperature. Should we spin the sample before freezing the specimen, or can we, do we need to add protease inhibitors to the sample? So we have done a thorough investigation on at least 6 biomarkers, which I would say KIM1, NGAL, cystatin C, cholesterol, and serum creatinine. And so what we have done is we done effect of freeze-thaw. So we did 6 freeze-thaw cycles, and we measured the stability of the biomarkers. And interestingly, most of these biomarkers are pretty stable, other than NGAL, which lost almost 20% of this activity after multiple freeze-thaw cycles. And also stability at room temperature. Surprisingly, KIM1 is stable up to 4 days at room temperature, and most of these biomarkers are stable for 24 hours at 4 degrees and up to 1 year at -80. These are mouse markers. Again, in human, we measure up to 4 years, and we found a 30% decrease in NGAL and almost 10% decrease in KIM1 values. So these biomarkers are pretty stable over a period.
Okay. Our next question, I believe, is for you, Dr. Sabbisetti, also. It says, why are there the different panels, and how do they differ in terms of detecting kidney injury?
So that's a very nice question. Actually, these panels are mainly based upon their dynamic range rather than actually checking the individual cell type of the kidney. That's what I believe it is. And for instance, if you look at the presentation, KIM1 is mainly you're looking at the proximal tubular epithelial cells, and whereas IP10 is a marker of immune system reflecting the kidney injury. So you will see IP10 if there is an inflammation. And VEGF, you see it because of the vascular damage that occurs in the kidney injury. And beta-2 microglobulin, again, is a marker for both proximal and glomerular injury. So these panels give a nice idea of ongoing injury status. And IP10 has been shown to be a very good marker in acute kidney injury setting. And KIM1 has been shown to be a very good marker in both acute and chronic kidney disease setting. So this definitely provides some mechanistic insight into the ongoing injury process. And again, the serum, the difference between serum panels and urine panels, maybe there could be some change in technology, but primarily it is, again, based on the dynamic range, because the dynamic range of the biomarkers in the urine significantly varies from the dynamic range of the markers in the serum. So maybe some of them actually can be mix and match. So it all depends what you're really looking at, what kind of injury is it, and is it primarily glomerular injury, is it interstitial injury. So based on which one can choose the markers that may actually reflect injury type.
All right. We're just about up to the hour, but we have so many great questions that I'd like to go a few minutes over, if that's okay with everybody.
Oh, that's fine.
The next question, I believe, is also for Dr. Sabbisetti. It says, is normalization to urinary creatinine necessary for the urinary biomarkers?
Well, that's a very important question. So the urinary levels of biomarkers actually change depending upon the hydration status of the animal or the patient. So basically, if you drink a lot of water, then you excrete a lot of urine, so the biomarker may be diluted. So in order to reflect the changes in the volume status, it is routinely used to people routinely use normalization to creatinine. Again, here people consider that the secretion of creatinine is pretty stable. So by normalizing to the creatinine, one can eliminate the variations in the biomarker excretion rate. Although in acute kidney injury setting, there are some studies in which shown that it is the changes in the serum, changes in the urinary creatinine are not stable, so they may exaggerate or undermine the value of the biomarker. So it is, especially in the AKI setting, it's always better to measure both absolute and urinary creatinine normalized biomarkers to make the decision.
Okay, the next question is for Dr. McGill, and it actually looks to be a three-part question. I'll read the whole thing, and then we can go back and tackle it one at a time here. It says, okay. What is the dose of acetaminophen used to induce liver injury in the mouse? That's the first one. The second is, and what is this kind of injury, whether it be cirrhosis or fibrosis or hepatocellular carcinoma? And the third one is, what are the humane endpoints that I should consider when inducing liver injury in rats? Okay, so back to the first one. What is the dose of acetaminophen that you use to induce liver injury in the mice?
So in our case, we actually fast the mice overnight beforehand. And the reason that we do this is to reduce variation in the animals, so we don't have to use as many. Because what we find is that if the mice are fed, there's quite a bit of variation. Even if you use a very high dose, some of them will not experience very much liver damage. So typically with the fasted mice, we can actually use as little as 150 mg per kilogram, which is actually very similar to humans. That's about the dose that both mice and humans begin to develop significant liver injury. That's the lowest dose to achieve toxicity. But what we typically use, and I think what most other labs who use this model also use, is a 300 milligram per kilogram dose, just dissolved in saline. It's very important in acetaminophen studies to consider anything that may interfere with cytochrome P450-mediated conversion of the parent compound into its reactive metabolite, because all of the toxicity is dependent upon that metabolite. So you don't want to use a vehicle like DMSO or polyethylene glycol that's known to competitively inhibit P450-mediated metabolism. So yeah, you dissolve it in saline, which requires a little bit of heating, and then we typically give a 300 mg per kilogram dose in the mice.
Okay, the second part of this question is, what is the injury that's actually induced, whether it be cirrhosis or fibrosis or the hepatocellular carcinoma?
It's actually none of those. So the injury that is caused by acetaminophen in mice and in humans is not a chronic liver injury. It's actually it's just acute liver injury. So you get hepatocellular necrosis, but it's a situation that resolves as the immune system kind of clears out the dead cells, and then you get proliferation of surrounding hepatocytes to fill in the empty space, so to speak. So it's a very acute form of liver injury.
Okay, and the final part of this question is, what are the humane endpoints that should be considered when inducing liver injury to rats? The question says rats, but I guess you can specify what you're going to be addressing.
So we've used both rats and mice, actually, and what we found is that rats are extremely resistant to acetaminophen hepatotoxicity. And this is actually consistent with what other people have found. So if we give a rat a dose of acetaminophen as high as 1 or 1.5 grams per kilogram, 10 times the toxic dose in a mouse, the rats will just barely begin to show a little bit of liver injury, and even then it's pretty modest. So it's better to use the mouse model if you're really interested in studying acetaminophen toxicity. That said, the main endpoints you would want to look for, I guess if the question is in terms of measuring the liver injury, my response would be you want to look at ALT, alanine aminotransferase levels in the serum, as well as perform H&E and histology to assess the percentage of necrosis. Or are you referring to other endpoints? Is it possible to find out?
I would need to look to see if they can provide follow-up questions.
Okay.
But for now, I guess we can go to the next question that we have. I believe this is for Dr. Sabbisetti. It says, what are the reference values for serum creatinine in mice, and what are the best lab methods for measuring kidney injury in the mouse?
Okay, so that's a very important question. I think, again, the reference level of serum creatinine in mice. In normal mouse, we will see the levels of serum creatinine in the range of 0 to 0.3. So, and we consider anything above 0.4, there is some injury to the kidney. So that is one. And so the reference levels are not really established, but this comes from our experience over the period and measuring serum creatinine using variety of methods, using HPLC, mass spec, or Jaffe-based assay. And to answer our second question, which biomarkers are better, that depends on the type of the injury you're looking at. If you are looking at proximal tubular injury, you are giving cisplatinum, and which is specifically induced proximal tubule. Then I would look into the markers for proximal tubule, so which comes early. And also the kinetics of these markers are quite variable. For instance, NGAL comes quite early, within 3 hours of the injury of ischemic perfusion or cisplatinum, whereas KIM1 takes 6 hours. And also the depending upon the type of the or degree of the injury, we have done 10 minutes or 20 minutes or 30 minutes ischemic perfusion, and serum creatinine, for instance, can't able to identify the animals with 10 and 20 minutes ischemia, but it almost performed equally to the KIM1 or NGAL in identifying the animals which are 30 minutes ischemia. So based upon the degree of the insult as well as the type of injury, the biomarkers, one can choose variety of biomarkers. But I would suggest to go with KIM1 and NGAL and serum creatinine, these three, because in most of the cases these three would be able to identify the injury at early stages.
Okay, I think we can handle probably a couple more questions here, even though we have a lot of fantastic questions still to be answered. This next one, I believe, is for Dr. McGill. It says, how exactly do you usually measure mitochondrial DNA?
So I actually do this with a quantitative real-time PCR approach. So I isolate mitochondria fractions from cultured human cells, typically cultured human hepatocytes, and then isolate total DNA from those mitochondrial fractions. And then I measure the total DNA using a nanodrop instrument. It's basically just the typical absorbance assay for nucleic acids, and then create a standard curve of mitochondrial DNA. And I do it that way because mitochondrial DNA is not widely commercially available. And then basically use that standard curve of CT values to calculate the concentration in my samples.
Okay, this one I believe is for Dr. Sabbisetti. It says, do you know if anyone has looked at plasma or urinary RNA as a biomarker of kidney injury?
Oh, yes. There are studies in which people have looked at the RNAs in the urine as a biomarkers. Actually, more and more studies are coming up recently to see RNAs as a biomarker in the kidney, but none of the names are actually ringing bell in my mind. But similar to microRNAs, a lot of research groups have been focusing on urine levels of RNAs. Definitely, yes.
All right. Well, we are not only out of time, but we're over time. So I suppose we should conclude today's question and answer session. As I mentioned earlier, we have a special offer for today's webinar attendees. We have a very short survey asking you to rate today's webinar, and when you submit your survey responses, you will be able to download a complimentary copy of EMD Millipore's white paper on a study employing Luminex-based technology to assess the ability of certain nephrotoxicity biomarkers to detect renal damage in rats treated with vancomycin to induce toxicity. So please stay on this page that you're looking at, and you'll be redirected to the survey in just a moment. Today's webinar has been recorded and will be available for viewing in the next few days. We will send you an email with details on how to access the recorded webinar, along with a PDF of the slides and instructions on how to personalize and print a certificate of attendance. So on behalf of our speakers and from me, Gwen Taylor, and our sponsor, EMD Millipore, we sincerely appreciate your attending today's webinar and hope you've learned some very valuable information. This concludes today's webinar, and we look forward to your attendance at future events from Current Protocols.